There was, I mean, I didn't know. They had like a, what, from like, I don't know, he was like a game show host in the 80s and 90s. And he has like, really about OCD and he was like talking about his experience with it and stuff. But like in the morning, they had speakers on like therapies. So they did, there was one talk on psilocybin, one on LSD, one on OCD. Yeah, and then one on ketamine.
Was it from Yale? Was it oh, what's his name? There's like a group that I know.
The famous guy? No, no,
I don't think so.
you probably see if it's on the blog.
Okay, good. All right, this is sharing. All right, so people who aren't here should be able to have this recorded. This particular lecture has not been previously recorded, so I'll share it with Dr. Julson to add to the GitHub. And depending if we get through everything for today, we can talk about brain clearing, but there is a recorded lecture on brain clearing that is available.
Just went black.
The screen, it was, it was Dale.
All good?
Yes.
Okay, so let me just check. I have no way to really independently verify if the volume's working, but I think it is.
I told you about that.
Yeah, she was talking about, and she's here.
Is it, oh, who's it? Is there some people?
Okay. Ah, I see. All right, so this should be recorded.
Sarah Jean Ferretti.
Okay, so we'll go ahead and get started since it's a little late and just try to get through this. And if we don't get to brain clearing, there is a lecture recorded already. So today we're going to talk about experimental design, especially with considerations of either recording or manipulating neural activity, which should be highly relevant for your presentations. It's also highly relevant for any grant writing, as well as just generally developing your PhD project. Okay. All right. So as usual, I like to start with a joke. So here's some comics. I usually don't volunteer for experiments, but I'm kind of a puzzle freak. That's one rat. And then some other mice are discussing. Of course, I'm glad we escaped. I just wish I could say goodbye to the cocaine button one more time. So the content in this lecture was pulled from a couple different sources. And if this is something that's really relevant for your projects, I would encourage you to go into more depth with these resources. So one is a course from Neuronline, which is available through the Society for Neuroscience. It's an optogenetics training series. I guess this cord is not so great. Maybe I'll try the HDMI link. Maybe that would be better. So now I have to reshare my screen. Working? Okay. All right. Yes. Zoom. Yes, I need to reshare. Share screen. Screen 2. Share. Okay, are we on? All right. And then the other place that some of this information comes from is Optogenetics Research Center at Stanford. So both of those are freely available and you can walk through the content. And so today we'll be discussing experimental design. So what are common designs when behavior is a big component? Because that's sort of the point of this class, behaving animals. What are we doing with behavior as a readout? Considerations for pitfalls that lead to challenges for interpreting and reproducing data, which is something we do want included in the presentations, challenges, pitfalls, etc. And understanding sources of variability. Of course, that's kind of the whole purpose of this methods class, is where is that variability coming from? How can you control for it and avoid it? And these can be from technical aspects of the actual manipulations and recordings, or it can be biological due to the behavior. So we'll talk about that and how to kind of discuss it as you're writing proposals or giving presentations. And so, of course, in the ideal world, and when you write a grant, you're sort of going through the scientific method of, okay, I'm kind of interested in this topic. I'm kind of wondering how this behavior is controlled. Here's some background information, you know, going from like really broad generalizations down to some more specific information. And from that information, you develop a hypothesis. And of course, this is all ideal. So you will develop a hypothesis. You will try to figure out ways to test that hypothesis with a set of experiments. You analyze the results, and then you either reject or accept your hypothesis. And if you have to reject it, you have to go back and make a new hypothesis. And in fact, the way people sort of approach this process is they kind of don't even articulate a hypothesis, right? You don't even, at the end, you know, whenever you go to seminars, it's like the end of the presentation when they tell you, now we have this work, after 10 years of research, we now have a working hypothesis, right? So it just depends how, you know, that's sort of how you actually discuss it with colleagues, is like, oh, we did all this work just based on a series of questions. And then after getting all this like firm results, we have a hypothesis that we know is right. Now this hypothesis is really true. But in fact, you have to, when you're writing a grant, especially before you have a lot of preliminary data, you have to kind of construct a hypothesis. And so it's actually harder to construct it out of kind of thin air and background until you have those data. So you kind of have to sort of think about hypotheses, you know, what are kind of the downfalls of them? If you had data, what would you be able to say? And that's sort of where you have to approach like an F31 when you're first coming in, you don't really have data on a project. The hypothesis could very well be wrong. You have to design experiments that will actually tell you if it's right or wrong. So steps to follow for designing an experiment. You have to form a specific and testable hypothesis. We'll talk about what that really means. In order to be able to know if whatever you're testing has an effect, you really have to have the appropriate controls. So we'll talk about that. You have to have a well-powered study. So we're going to talk about a little bit of how you determine sample sizes for these kinds of experiments. And then you need to execute it rigorously. And so in the case of a grant, you have to include some details that might seem very obvious to you as a person who is intelligent and rational and has done experiments, but a grant reviewer will assume that you are not intelligent and you're not rational, and maybe you've never touched a pipette or mouse or anything. So there are some things that you have to say to communicate those things implicitly as you're writing it. And then really digging into how you analyze and interpret. And so this seems really overwhelming to fit into, let's say it's an F31 into 6 pages. And so I'm trying to give you all these sort of bullets so that you really figure out how to communicate these things in the most efficient manner so that you can get it into those 6 pages that you need to, or in the case of your presentations, into a short amount of time. And so there are ways to really be able to communicate this just in the way that you present the information so that it's all built in there already. And again, this is all supposed to be really helpful to you, you know, in your PhD process. Generally, in writing an F31, you need to talk about the significance of the problem, communicate what that hypothesis is in a specific testable way, say why it's innovative compared to, you know, this again goes back to what has maybe been done in the field, how you're approaching it in a different way. And then in the approach, which is really kind of what this class is about, is really nailing down, you know, approaches that are solid. So experimental design, including sample sizes, controls, outcome measures, rigor and reproducibility, analysis details, which we'll talk about today, and the ways that you can interpret and thinking about caveats. And we're going to talk about a lot of technical caveats today. A lot of times in a grant, or even in this presentation, you'll find caveats. You might put like one or two technical. Nobody really can, as much as they think you don't know how to design an experiment, and you have to sort of hedge on that when you're explaining your experimental design, when it comes to the pitfalls and alternatives, nobody cares about the technical pitfalls. They all kind of know. They really want you to talk about the sort of stuff that's arising from biological consequences rather than the technical. So you can mention a couple really briefly, but you definitely want to address what are alternative interpretations when you get data. So the first thing we'll talk about is developing a specific and testable hypothesis. So in the case of like manipulations, and we were talking about neural activity, a lot of times this is sort of, are you testing necessity? Are you gonna like inhibit neurons? Are you gonna knock down a gene? Something like this. Or are you trying to activate during different components of your behavior or test? Are you doing both? Are you trying to activate and inhibit? And what might the differences in interpretations be for those things? So what aspect of behavior is your target neurons, or brain region involved in? Actually change this from the original presentation to be more inclusive, because some animals don't have like brain regions. So all of this information could still be applied to C. elegans, where there's a neuron that does something. So what aspect specifically is it involved in, and what might it not be involved in? So you can design those control experiments. And when during behavior do you want to manipulate? Do you want to activate or inhibit when activities really critical during the task? Do you want to manipulate the activity throughout some sort of long naturalistic interaction? Do you want to actually change the window of activity or inhibition? So these are things that we will discuss, the nuts and bolts of later. And so is there a specific cell type of interest? Are you kind of just inactivating or activating a whole brain region? Is it types of, is it excitatory projection neurons, local inhibitory neurons, a large population, a specific subtype, deeper superficial layers of it? Like if there's some laminar organization, like where specifically are you trying to target? One side of the brain, both sides of the brain? Is it something that's lateralized or maybe not lateralized? And are there physiological or molecular features of the cells that are already known that you can take advantage of to kind of narrow down some of these questions? And so this is just like kind of me thinking of all the different things. It's not exhaustive, but it's just kind of as you're thinking through, these are the kinds of things that you might want to be specific about, because it's what people will think about. So we'll kind of go in a little bit of an order, but it might not be like a super great order. But just first, I kind of wanted to introduce you to sort of what is it you've probably already encountered in journal clubs or in seminars, what typical kind of experimental designs there are. So there's typically, in a lot of rodent studies, there's sort of the time that you inject viruses, maybe implant electrodes or fibers, things like that. So you do your viral injections, and they say, and I know a lot of people do not do, I personally have never, except for one time when someone else was giving me the virus, which was when I had there were 20 techs in the lab for some reason. So someone was there to just hand me a virus A or virus B, and I injected. But almost never do you actually alternate YFP and Halo, and then go back to YFP, and then go back to Halo, for example, while you're doing a session. But what's critical is to not just do, okay, in this one whole experiment, I did all chanorodopsin. And then I went back 4 months later, non-litter mate animals, and then I did my whole control experiment, different time of year. Someone else was helping me with it. So the idea is at least, let's on each surgery day, do a couple YFP, do a couple Halo, so that it's not like, there's not big batch effects. And then, with some of these learning paradigms, you might be training animals on a task for several days, well after this surgery. And then in the last, session, you might excite or inhibit. And then the typical outcome measure, if you are looking at, let's say, learning, you're gonna have some percent successful trials, and you want to make sure that both groups are kind of learning if you're not manipulating. So again, balancing groups by age and sex, alternating your viruses so that you have balanced groups whenever you're doing testing days, to eliminate any batch effects, and make sure that the experimenter who's rating everything is blinded to the condition. So they don't know if they're rating behavior of a YFP animal or a halorhodopsin animal. And in, maybe next year I'll know something about machine learning and be able to teach it, but at this point I don't, but know enough to give a whole lecture. But if there's subtraction of humans from the process, maybe you don't need to account for bias. Everything's automatically scored. But there's still some question about, do people run animals differently if they know that they're, one or other. So just being able to reduce bias. So this is something you definitely want to include in a grant, something about experimental blinding. And again, that's just two words at the end of like experimental design, experimenter blinded to condition. So that's pretty simple to incorporate. And then during the actual testing, you want to reduce variability by habituating animals to the laser tether, if that's the case, habituating to the behavior room or to the apparatus. If you're using optogenetics, you want to be able to obscure the light from the animals. There used to be I don't think I have the picture in here, but there was this animal with this like blue lightning bolt coming out of its head. And they're like, ooh, optogenetics. It's like, well, the mouse can see that. You don't really want them to know when the laser's on or have their behavior affected by the fact that they have this lightning bolt coming into their cage. So you can block the light coming out of the patch cord. If you're using chemogenetics or something, you definitely want vehicle control, adapt animals to being injected. We'll talk about exactly why you need to use the absolute minimal power for the stimulation of your opsin. And then you can also consider using a redshifted opsin, which are typically more sensitive and you can use even less power and there's less artifacts from the light. And we'll talk about what those artifacts are. And then of course, this design between YFP and halo, typically you would have laser on and laser off for both. So it's not just like laser on with YFP, laser on with halo, you would do both conditions. And then you can really see dissociate the effects of any artifacts due to the light delivery from anything that's actually due to activation of the cells. Is there too much problems? And so in that case, if you do laser on, laser off, and you have YFP and halo, that is between subjects and within subjects design. So these are buzzwords, keywords that you can just say, and then people know what you're talking about. So you don't have to get like you don't have to put this huge cartoon of like exactly what you're doing. If you use those words, people know. So again, this is the between groups. So in this case, it's totally separate animals, right? You don't have YFP and halo in the same. So, hopefully something happens, you inhibit cells, and there's a big difference in the YFP and halo. And then also you can have within subjects. So here again, you have laser off, hopefully they're the same, laser on, hopefully the YFP guys don't change because of the laser. That's not always the case. And then you have a big difference. So in this case, it's within and between subjects. And because you have repeated measures with different groups, the other buzzword that you need is randomized crossover counterbalance design. So technically these kind of mean the same things, but again, you're going to be assumed that you don't know to do this and that you will do it wrong. So you need to expressly say, I'm going to use randomize. What does that mean? I'm not going to say all the animals get off first and all the and I'm telling you this because everyone does it wrong. Everyone's like, well, I need to do the control condition first. What you really should do is have a group where you've got about 10 YFP and 10 halo, and you give 5 laser on first and 5 laser off first for each of those 2 groups. And then on the second test, switch it. And so that's what it means by crossover. It means now this one went from on to off and that one went from off to on. So that's what it means to crossover. And counterbalance means it was split. So not all of them got on first, not all of them got off first. So they were randomly assigned to one condition first. You didn't pick and choose based on their past performance on this learning task who got on or off first. So it was random. I drew a number out of a hat and then I counterbalanced it by switching it.
With crossover, how exactly do you decide what animals go in each group? Like, I'm used to with clinical trials, like there's like an algorithm to doing that, but with mice, how you decide, like how you actually get them separated into their 2 groups.
So you should not be deciding. That's the whole point. It should be totally random. So you should have. So whoever is assigning the group, this is where blinding comes in. So I have n equals 10 YFP, n equal 10 what halo. So I go, now I have mouse 1, 2, and then it goes to like 20. So that's how many we have, right? So ideally, the person who's running the behavior only knows 1 through 20. Someone else went and made sure that between the halorhodopsin, which there are 10 of, and the YFP, which there are also 10 of, they got randomly assigned. So what I have done in the past is used a random number generator. That's just like, oh, gives me a 0 or a 1, and that's like laser on or laser off. But then you also have to counter, you can't just, even if it's like, if it's random and the only choice is on or off, it could give you off for all of them. So it has to be balanced in the sense that half of these guys need to be laser on, half need to be a laser off. So that I don't have a big, like, oh, it could be that, you know, if it's a learning task, they performed a certain way just because it was the first time, and then you had a second time. So you have to be able to dissociate the effect of the laser from the effect of the session day.
And does that take into account like the weight of the mouse or like any other factors about the mouse other than just doing it completely random like that?
So you probably don't want to take into account the behavioral outcome for sure. So weights, that might be something that's really dependent on your outcome measure, but you should have, this is where counterbalancing of may come into play of like, I want to make sure they're the same, but if you test them twice, like the fact that they're a bigger mouse, it shouldn't matter if the laser was on or you see what I'm saying? So the randomization shouldn't necessarily care about that because you are still going to collect the measure on 2 sessions. And maybe even 4 or 5, like you can keep going back and forth if you want a lot of data points. But you know, at least in a case where there's 2, you need to make sure that there's counterbalancing, which takes into account the fact that like I really need half of these animals to be one or the other, but otherwise kind of, so it's like semi-random, right? So it can't be completely random, otherwise you might end up with the same issue. And so, what is it that the across groups will tell you? It will tell you if there's something weird about one of the viruses. Is the fact that there is a halorhodopsin in there going to be something that's changing how the cell is even without light? Light delivery. So of course, you know, hopefully you're getting some change with halorhodopsin, but you do need to shine the light in the YFP case to see if there's any effect, to be able to dissociate the fact that it's, you know, changing membrane properties versus just receiving light. the insertion of the fiber and cable. You can't just have a YFP animal that just runs around. It also needs a fiber. It's gonna get hooked up to the light. It's gonna get light delivery. And then behavior training and exposure, they should be going through the same number of trials, the same recording sessions, everything. And then the other design, again, we're going to talk about some of the artifacts that can arise because of heat and light from lasers. So this is kind of specific to optogenetics. And then also when you have repeated measures, if you have this crossover design, this can increase your power. So you should, if you input these parameters into a power calculator, you should need fewer animals for the experiment if you do repeated measures. And so depending on, what you're trying to do, if you're trying to do just a couple sessions, if you're trying to do multiple stimulations, there are different patterns of stimulation you can give. You can have an interleave pattern within a session where you have kind of a baseline and you stimulate, then it goes off, or baseline inhibit off. You can reverse it depending on what you think the laser is going to be doing. But this way you have repeated measures within the trial. And again, you can have them inverted, depending on what it is that you're trying to do. And again, you can have it where you're doing on off in quick succession, because you have say a 2-minute trial, there's a cue during 2 seconds and you just want to stimulate during that cue. You can have some animals where they get it during the first cue and then randomly change it on random or alternate, whatever it is, but you can randomly assign them to have off first, then on, and then repeat the pattern, or have it on first and then repeat the pattern. With inhibition, typically inhibition is given throughout a session, and I'll tell you a little bit about why that's probably going to be the case, but a little bit it has to do with the biophysics. You all talked about biophysics of Halorhodopsin and arch. So I'll just show you a couple pieces of information that maybe you already saw, but just to show you, in the sense of experimental design, why you might have the laser on. You might even have it on before you start your behavior so that the cells are inhibited before they even start some kind of behavior task. You can also do sort of random inhibitions or activations. Then there's open loop activation. And again, here you're just sort of I'm going to have the laser on for 5 minutes. Sorry, that would be way too long. If you're having chanorodopsin, have, 5 seconds where it's on, 5 seconds where it's off. And then I'm going to segment the video and see if there's big differences between those time periods where the laser's on or off. You can also now, with the advent of video tracking software, you can actually say, hey, I want to analyze what these animals are doing in real time, such that, let's say they're encountering an aggressive mouse. Anytime they escape that aggressive mouse, I want to turn the laser on. And so you have some sort of DLC, which is deep lab cut, or sleep. Forgot what it stands for, but it's not SLEEP, it's SLEAP. But you have real-time pose tracking. You can feed that back into a class. So you need a very powerful computer, by the way. You track them, get an algorithm that's classifying that behavior, and then triggering your laser so that every time they escape, they get a light pulse. So that's considered closed loop. And again, that's because a loop is involved. You can also have human triggered closed loop, which is what I have experience with. You just sit there and watch them, and every time they do something, press the laser, and then it'll go on for 2 seconds. So that's, if you don't have computer vision, you can use human vision to close your loop. And so a lot of times people actually if these are compared. So having open loop, just a randomly delivered laser, versus what happens when you specifically deliver. And there are some really good papers recently that have shown that there are cool differences when you do open loop versus closed loop. And then of course, for excitation especially, you might want specific patterns of laser delivery. So of course, I'm sure you've already talked a little bit about, what is hertz? That's like how much second you have of delivery. So 2 hertz versus 5, 10, 20 hertz. And these are all sort of in the biological range of what you can measure from a neuron. So you might want to give different stimulations. So in some research that I was involved in a few years ago, we were actually doing projection stimulation to different projections from the hypothalamus. And we gave these different pulse hertz of frequency. And so one interesting thing that we found is like at a pretty typical 10 hertz stimulation, which is actually what I used in the behavioral assessment of these animals. I was doing 10 hertz with the infant-directed behavior. We could see that in all cases, we had a really strong response physiologically with the first pulse, and then sometimes you didn't get another pulse after that. And that's due to the actual properties of each different projection. So you also can't necessarily assume that whatever you're doing with the light pattern is being followed by your cells of interest. And this kind of played out in the behavior where we saw different effects of this 10 hertz stimulation in different projections. So moving on now to any questions about just setting up the experiment?
For the last slide, are there like I know we talked about like 10 hertz, 20 hertz, like is there for standard octo, like a number that you regularly do, or what does it depend on?
What you typically see for most cells, so there's going to be a little bit of a difference in what you want to do at soma versus projections. And I'll show you some data from papers showing the difference between soma and projection stimulation, how much power you need. I'm not sure that one actually looked at the frequency, but it's generally thought that like soma are going to express more of the channel, and they're going to be fewer at the terminals. But then the difference also is that the soma, usually I see somal activation at 20 hertz and projection at 10 hertz. But all of this has to be taken with the caveat that a lot of early studies, and it's still kind of early days, at least in terms of actually going in and doing some of these controls in your samples, were from like cortex, where you have really specific cortex and hippocampus, where you have sort of pyramidal excitatory type of cells. And so then when you actually get in and do these control experiments in your own system, you might actually have been better off doing a different, like you have to check. So this can be really specific to the cells. So, you want to kind of look in the literature or do these control experiments yourself, build them in to say like, oh, I'm going to check and see what my cells can actually follow, and then use that pattern of stimulation. So that's a big kind of caveat in the field. And that's also why people like to see excitation and inhibition, because if you did some kind of weird supernormal stimulus on your cells that they can't either they go into depolarization block, which I can show you. So you're actually only seeing activation early, and then they actually inhibit. And so you're not stimulating them the whole time. So that's why it's good to compare it to inhibition, because in the inhibition case, you kind of just subtract that firing from the system. And so you may have a weaker effect or a slightly different behavioral effect of inhibiting. And that might be more in line with the natural, like you've deleted the natural firing. Whereas when you activate, unless you went and did these this is in slice physiological controls or recorded while you were stimulating, you don't know if your cells were following that pattern that you were delivering. I just got excited. Any other questions? Okay. So This is always important to say in especially a grant proposal, but also, you know, if you're ever giving just whips or talking to your committee, they might be interested in what kind of sample size you're looking for. And I will tell you right now, the answer of I'm going to like a P that's significant is not a good answer. So have some sort of so the point of doing a power calculation is that you have an a priori idea of what the target is. And hopefully to avoid batch effects, you already pre-plan how many you're going to do. Like I'm going to have, this is the pilot study. I'm going to have a balanced control group, 5 halo, 5 YFP. Then 2 weeks later, I'm going to add, you know, another set to that. So a good design has high statistical power, and this can be calculated. What is power? Is the probability that your test will be able to detect a significant difference. And that difference depends on how big your effect is. So effect size is something that also, you know, it ties into this power calculation. So the best way to estimate a group size and the power that you want to achieve is over 0.8. And I don't exactly know why that's such a, that everybody wants the best power possible. 0.9 is great. You can't really get 100%, right? You can get 100% power. So 0.9 is like pretty great. 0.8 is acceptable. 0.7 is starting to be iffy. You know, human studies are like 0.6, but you know, it's humans, so people care more, so they can get away with more. So I'm being a little facetious, but these are based on things that I've actually read in papers. So how do you perform this power calculation? You need to have some kind of information, and some of it can be guesses as long as you explain that stuff when you're putting in the statistics section. So the mean and standard deviation of your control and your manipulated group. And so that gives you the effect size. And effect size is, it can be measured in a couple of ways. One of them is Cohen's d, but essentially it's not just that there's a, you know, let's say chance level is 50%. And when I stimulated, I got down to 47%, but it was, the data was so tight that it was significantly different, but that's effect size is 0.03. So it's not a very big effect size. So people really like to see huge effect size, even if the deviation is a little bigger. So like something like 30% or larger. So effect sizes are considered to be large if they are like 0.4. So that's like 40% of variation is explained by this change, and there's like this bunch of difference. Smaller effect sizes are more on the order of like 10%. So that's a small effect size. But again, depending on the biological measure that is on, you know, your y-axis, that can be a big difference. Like a 10% change in synaptic protein can be biologically meaningful. So, but you might have to have a larger N to be able to say that that is a significant difference. So p-values are known as alpha. The power is known as beta. Again, 0.8 to 0.9 is what you want to see. And I can load these slides, but these are hyperlinks to places where you can calculate these.
So in terms of like making sure you have a high power, would you have to like essentially do a cohort of like do an experiment and then figure out the power and then say, okay, I need to redo it because the power is too low?
So yeah, the way that people usually write, and again, it's just a few sentences and it just has to have the like right words inside of the sentence. So what effect sizes can be based on. So if you're doing something really standard, like fear conditioning, there's usually published data about, you know, what are the effect sizes with fear training. You know, you may have a hypothesis where you're looking at a specific pathway. So if you want to say, I anticipate that the effect size is going to be this, either based on previous data that looked at a similar pathway, so there's published data of an effect size, which is not the very best, but it's a place to estimate. And if people aren't reading carefully, they're going like, oh, it was based on something. So there was some rationale behind that effect size, or you have pilot effect size. So in pilot data, you can estimate an effect size even if you don't have but like an N of 2. So even though you can't calculate a p-value, you can estimate an effect size. Say like, well, I did an N of 1 and it was a huge difference. Like it's, you know, it's pilot data. So that's fair to say. Again, the point isn't that it's like publishable in when you're, you know, making a grant, or if you're talking about pilot data, but you could say, you know, always hem and haw, and you say, well, the power's not there yet, but this is the effect size is looking like it's going to be this, and I will need this many more to be able to reach significance. And so if the effect size holds, you will reach significance if you use the number of mice based on that power calculation.
Okay, so you usually just like do research before you start the experiment to see if you can find like an effect size or an N that give you the right power?
Yeah, some people, sorry, I meant to also say, you know, you either have pilot data or you say, you know, we've been studying this pathway in other behaviors and this pathway has an effect size of this. So, you know, your own past research, your experience with these manipulation methods. So usually there's some sort of hand wavy thing of like, oh, based on past studies, you know, 37 through 39, reference 37 in our own experience with these techniques, 40 through 45, we anticipate an effect size of blah. So we're going to use this many mice to obtain sufficient power, 0.8 or higher. Yeah, so that's like the sentences. So, and again, the statistics part is really like small, but it is the sort of thing that can like make or break a proposal. It can make or break a grant. It's the kind of thing that people can get picky with. If you're giving a presentation, your committees are going to want to know you did it. So just know what all those phrases mean for those two sentences. Yeah.
So I'm taking like this clinical design course right now too. So we clearly just covered this, but like with human subjects, obviously, and they like make you like have a meeting with like a statistician. We don't have to do that, or is it like normal for researchers to have like a meeting with a statistician as well to like get your sample size?
I think I have information about our biostats core, but in a different part of this. I don't think I said it yet, right? Yeah, so this is more about like power. So I showed you the experimental design and like how to decide like based on, you know, you're in the lab, you're kind of the biologist person. So statistics people are going to have a strong opinion about how you should be designing it based on numbers, purely numbers. So there has to be some kind of like coming together. So one reason why, you know, your qual is usually really helpful, even if it's in, I heard some talk about qual practice, but it's intimidating and it's going to be tough. And like, again, don't be married to any hypotheses or like, you know, experimental designs until it gets like skewered by people. But this is the kind of thing that typically will get raised. So your experimental design, as I mentioned, it directly, if you have repeated measures, it changes what you need to achieve a certain power. And so the power can also be affected by your experimental design, and a statistician can help you. And people who have been in the field, like people on your committee, qual committee or student advisory committee, they have experience with this. You certainly can meet with a statistician. In advance is better because it can be very frustrating if you have already collected data and they say, well, you did the design totally wrong. So, you know, it's unanalyzable, which isn't always the case. Usually there's some design that went into it, but it is a difficult conversation sometimes with statisticians, but we do have a biostatistics core that can give you input on design. And that's part of why I wanted this to be part of this class so that you know some of the parameters that you want to think before you start. Because again, there's some really easy traps to fall into of like, oh, I'm going to do all the animals with laser off. And then I'm going to warm up because you have to warm up the laser, like blah, blah, blah. And it's like, oh, well, then I'm going to do all this on, and 3 days later, I'm going to do all the animals on. And then you have issues, and then you have to go rerun the experiment in the reverse order on a new cohort of animals because you got excited and killed them, and now you don't have them anymore. So these are things that you really want to think about in advance. So if you can meet with the biostats core in advance, we do the computational department has that resource available, and I do it later in the presentation. I have their contact information. So just to summarize, of course, now you've randomized your group assignments, you've randomized how you've put the virus in, collected the data, the experimenter was blind to condition throughout collection and analysis. And then you actually this is what you're supposed to do, and I know people do, this is what you're supposed to do. Don't exclude anything before you know, like if you're gonna say something's an outlier, it should be an outlier among the entire group, not just the control group or the experimental group. So you should really be thinking in the most rigorous way to design it is to design exclusion criteria before you collect the data. When you collect the data, you know what you're throwing out before you even break the code of what goes in what group and plot it. So executing a rigorous experiment. You define in advance the power that you're trying to achieve. You design in advance the pattern of stimulations and the set of behavior tests you're going to be doing. Your control groups, evaluating the specificity of viral targeting. So this is something you want to be able to include, exclude before you break the code, section the brain, stain them. Was my fiber over the place where I thought the virus was supposed to be? Have a predetermined cutoff for what's sufficient injection. It shouldn't just be like, oh, well, I looked at the behavior data for this one. I looked at the virus injection. It was kind of low. I don't like that data. This is a good enough reason. Like, you should decide in advance. So predetermined cutoff for what's good infection. So within whatever millimeters of your desired site using stereotyped coordinates, histology, of your fluorophore placement. Somehow you've placed the fiber either by staining it with a dye or looking at DAPI damage. Where is the fiber implanted? Again, is it close enough? What is close enough to your target? What's on or off? Because a lot of people also, you can use data from, if there are animals with off target placement, you can actually put those in a control group. Still use that data. Look for possible toxicity. So viruses from AdGene are really high titer, which means that they express very highly in your cells and they could be killing your cells. So you want to be able to decide if there's toxicity. That can be abnormal morphology of infected cells, aggregation of the fluorophore, loss of fluorescence, where it looks like all the animals that got halorhodopsin, there were no cells infected, even though all the control, that means all of the cells maybe died. And maybe they died because they were infected with halorhodopsin, or maybe they died because of the laser. So you need to be able to assess that. And then behavioral data. So that can be like if you are recording some sort of predetermined control parameters, so quality assurance within your recording wasn't met, like the baseline was really weird, there was really high background activity, there was noise, or if you collect data and one of them is just really far out, you can use a Grubbs outlier test, or as you're making, what I like to, so I'm sure Dr. Jolson talked about how data sometimes doesn't get saved to the hard drive. Sometimes data doesn't acquire the way you thought it was going to the cloud either, or like something happened in transfer. So it's always good practice to make a note if you are like in the room, like, did that mouse's cable get stuck on something? That's something that might be outlier, or sorry, exclusion criteria. Did the mouse never enter the box, never nose poked, something like this that would be like really, they didn't basically perform the task at all. Do you have some kind of criteria for inclusion, exclusion based on task performance? And that can be assessed either again by notes, quality assurance, or if you actually have data and it's just really far outside the deviation. So GRUBS is a test that tells you if it's more than 2 standard deviations outside the mean. And I think GRUBS will use that cutoff and it can identify maybe more than one outlier, but there's some tests that only identify one, and again, like an outlier, like once you throw it out, it changes, the distribution of the data. And so generally speaking, there's only one, but GRUBS, I think, can identify more than one. So other considerations for rigorous experiments, consider how you're going to validate your manipulations. Again, this gets back to Emily's question. How do you decide what stimulation pattern you're going to give? Are you going to be validating what your optogenetic stimulation protocol does to physiology? Are you going to have probes that are recording while you're stimulating? So I just gave, some examples of in vivo, there's some stimulation that's happening with the optogenetics while there are neuropixel recordings. You can also go and slice later and check what was happening to the physiological properties. So that can validate if your construct is exciting when it's supposed to excite, because sometimes inhibitory constructs excite. So you have to be careful about that. Verifies the kinetics. Again, are you having depolarization events every time you stimulate? Is it only the first time? Do they, is there plasticity involved in this process? What kind of network effects? So you might be thinking, okay, well, I'm inhibiting, inhibitory interneurons, and it's just going to affect the local circuit. Well, if they have big effects on the excitatory properties of projection neurons from the cortex, you're going to have network effects. Those could be seizures, it could be synchronous neural activity that either within that brain region or between that region and another region. You can have disruptions of chloride reversals in the case that you're trying to do inhibition, post-inhibition rebound activity, which I have a slide on later. You can validate, again, I think I already said, but yeah, ex vivo slice physiology, what's going on with the kinetics. Is the pattern that you chose really something that the cells can follow, whether it is the cells of interest at the soma or if it's projections, are the, postsynaptic neurons able to follow whatever training you're giving? And I'll show you some examples of that. And then this part is pretty essential, and I think it's one of the pieces that maybe gets the most ignored. When we're telling you all about the techniques, we're telling you all about experimental design, we're showing you results from other people, and a lot of times the interpretation side is kind of left out because when you're discussing a paper that's published, it's really clear, but if you don't actually have results in hand, it's hard to understand what are the alternative interpretations before you really have data in hand. So for typical data analysis, between groups analysis, you would probably use a Wilcoxon rank sum test, and that's when you have data that's non-parametric, and again, it's between groups, so they are not matched observations, so it's YFP versus halo. Otherwise, you'd use an unpaired t-test, so that's if the measures are parametric with no matching. And how you would determine whether you do the Wilcoxon or the t-test is by using a Shapiro-Wilk normality test. And this can be used either for between or within group analysis. I just want to make sure to mention it here. So this you can run before you start doing any t-tests or anything. So that'll tell you, yes, it's normally distributed, and that means it follows the bell curve. But a lot of times, as you know, in the case that we were showing, like with percentage, correct being your parameter, you're going to have a skew in the data usually. So you can have a skew to the ceiling, which is everybody's at 100%, especially if one of your inclusion criteria is they have to be over 90%, then your control group's going to be 90%, like maybe plus or minus a little bit. So if you have a skew in the data, you're going to end up using the Wilcoxon rank sum. So that's kind of why I put it first. But if you have a bell curve distribution, you can use the unpaired t-test. And again, making that decision is if it passes or fails the Shapiro-Wilk test. So if you have repeated measures, so it's laser on off with just your channelrhodopsin group, you're going to use the Wilcoxon signed rank test if it's non-parametric. And if it's parametric, you use the paired t-test. And again, this will change the power level of the observations. Hopefully you need fewer. And then if you have mixed effects, which is if you have the channelrhodopsin and YFP group and you do laser on and off, that is known as mixed effects because you have both between groups and within groups. And so this is a really, I feel like there's a lot of shade thrown on Twitter about mixed effects, but that's essentially all it means is that you have some between groups effects and some within groups. So anytime you have repeated time points, it becomes mixed effects as long as you have a between groups or control. So again, that comes down to the intersectional design. Sometimes you have more effects. So mixed effects, I think, can take into account like, oh, I had to throw one animal out on one day. So it'll also tolerate if the repeated measures don't come from every single animal. Repeated ANOVA, two-way, there's three-way, there's nested ANOVAs. These can take into account a certain number of parameters, and it can take, it could be either two-way or two-way repeated, but the ANOVA really wants every measure perfect. So you can't throw anything out or have accidentally had a mouse die halfway through, which definitely happens. You can also do an ANOVA with mixed effects. And they become, the more complicated your experimental design is, the more complicated the statistics becomes. And so here's where I have the information about the biostatistics core. And so I kind of put this because a lot of times the statistics are what help you interpret your data. But yes, as we already discussed, you might want to already have a good design in hand before you go collecting data. For sure, with human data, you have to know in advance because you're probably just barely going to make it to getting enough data based on enrolling participants. So you really need to know people's time is precious. You usually have to pay for it. But the same is also true if you're in the lab, it's kind of your time that's going to be saved by getting a good design. And also getting the most data that you can out of the animals that you have. So being able to test, different laser conditions, closed loop versus open loop, repeated sessions. Maybe you want a training task that's for learning, but then you want some control behaviors like locomotion just to account for any variability. So what are caveats for interpretation? And again, so these, some of these are sort of the technical ones that reviewers are not going to care so much about when they look at your grant. So there's going to be like laser light or noise of the shutter. But again, if you designed your experiment a certain way, you're going to be able to account for that because you already have your nice rigorous within and between subjects design. But there's some that are kind of like, oh, well, you stimulated and they didn't quote unquote freeze as much in this test, but what if your stimulation caused them to walk more? And so they just don't freeze, even if there's no cue pairing or something like that. So in this case, you would do another behavioral test. And have no cue pairings or something in just an open field locomotor test. How long did the laser have an effect on locomotion? So that is a confound that is not so technical. It's actually something that would happen because of the stimulation you gave. And it would be able to really refine your interpretation of saying, not only does this affect learning, but it does not affect locomotion, so we can rule out locomotion effects. So that's really what people want you to do when you're giving caveats. It's not just to say, well, you know, we had our within, we're going to add a YFP group to make sure the laser doesn't do something. Nobody, people expect you to already design those technical concerns into the experiment. So then circuitry confounds. These are also things that people really want to see you address when you're talking about these things. So when you interpret, you say, oh, we inhibited and we got a loss of effect on this learning. And it's like, well, we can interpret that as being a direct effect of this projection, or alternatively, it could be a circuit level effect. So these are the kinds of things that people really want you to kind of think about when you are talking about alternative interpretations. So you have the one that's really directly related to your hypothesis. And even if that result supports your hypothesis, there could be other hypotheses that that same result would support. So that's really where they want you to be thinking about. We'll also talk about response latency. So, you know, I stimulated, but then there was this behavioral effect like 5 seconds later. So what's up with that? Again, that could be interpreted as some sort of like downstream projection, collaterals could be activated, something at the circuit level could be going on there. So certainly your manipulation did something, but it might not be related to the activity that you introduced. It might be some downstream activity. There's also complex neurochemistry. So a lot of times there's also, you know, to say, well, I excited, you know, dopamine neurons. Well, dopamine neurons notoriously have, you know, glutamate, they have GABA, they have other sort of neuropeptides that could be co-activated. So these are the kinds of things they really want you to think biologically rather than just purely technical. Because the technical stuff you should really be addressing with the design. Physiological confounds, again, we kind of talked about this. Are the neurons physiologically capable of following the train? Can your opsin itself follow the train? Or are you giving something that just that channel can't follow? And can your source of light follow the train? Because there's on and, you know, LEDs are much better at following the on and off. Lasers, you know, there's some like ramping up, ramping down that can happen, but most people are using LEDs these days. You need to kind of take a step back once you really get your results and think about the full picture of like, okay, this supports or doesn't support, what else might I be missing here?
I guess I'm just wondering, like, for these various compounds, like a lot of these can be, like, pretty much every experiment could be happening. So when you're writing about that in a grant, like in your fall, like, how are you supposed to say, okay, this could be happening, but we know that it's not happening? Like, you have to do an experiment for each thing to say, you know, it's not happening. But if you're doing like an experiment with dopamine and then you say, oh, these other neuropeptides could also be being activated and activating other circuits, like what else are you supposed to say?
So again, like part of the reason of discussing all of this is to give you kind of the full framework. And so a lot of times in grants, you're going to have, you know, sort of there's a big hypothesis, there's one or two aims, and within each aim, there's a few experiments. And so the idea is to kind of comprehensively cover these, but not necessarily for each experiment. So, you kind of use your real estate wisely in the sense of like, what's the most concerning thing if I'm, doing this particular sub experiment, and explain that. And then, if there's a concern that applies to multiple aims, you might say that. You say like, oh, this is a caveat. This applies to all these other sub aims. We've thought about it and we're gonna do this. And then that covers your bottom for the rest of the experiments. And you kind of do that for the rest of them. And maybe you don't have space to do all of them. You say another one in another sub aim, and then you refer to each time that might be the case. So again, you have to kind of know what all the possibilities are and be really judicious about when you're saying them. But just so you kind of are aware of those things, let your reviewer know that you're aware. And again, you can't keep restating the same stuff. So, sometimes you might sometimes what people do when they have like the approach section is they have a general methods part where they talk about that. And that's where they put the technical stuff. That's like, oh, this is a technical caveat. For that reason, we're designing it this way. And that applies to every single aim in the whole grant, and you don't keep saying it. And then there are some that are really specialized that you're going to go and put in those experiments. So you can't cover everything. You won't be able to, but the point is to let people know, your reviewer know, that you are aware of certain issues. And again, you can obfuscate the need to talk about technical caveats if the design is good and already takes those things into account. So what is optogenetics used for? I'm not going to get too much into chemogenetics. They're a little bit different sort of technical issues. So most of what I'm going to show you after this is opto examples, but you use it to manipulate behavior, test functional connectivity, combined with imaging or recording. And in some cases, it can be used for translational applications. So we'll take like 5-minute break, and then I'll go through some actual examples from the literature of all of these things. So you can see in practice how to apply these things. So we'll come back at 2:50. It's really hot in this room. It's really cold in the hall.
The like thermometer is like something with the actual like monitoring.
Are these also things that we'll talk about, like in wall class and stuff, where they say like, okay, you should have this sentence about statistical power in your grant or whatever, or that's like, is there another place we would learn that?
I honestly have never been part of a qual preparation group, so I have no idea what goes into it.
But like, is that
Do
something
you
we would get
remember?
for?
I think in the class, like the whole thing in the class, they do stuff.
Okay.
To improve these things.
Okay.
But honestly, I feel like a lot of this information, I gathered from what
Yeah.
like not explicitly taught, you know.
You're aware. Oh, yeah, it's the sevens, not the eights.
I'm so excited. I'm so
Yes. Hilarious. Gee, you do it. You're better than most. I'm better than most Americans. Oh, it's so funny. I need to tell you. I need to know what surveys. I find that hard to believe it. Like Bruce
My
Lee,
other roommates,
Lucy Liu. Yeah.
my other roommates, from South Carolina was.
Was a West Asian American. But it's just like most Americans. Can't be right. Where, what is that from? It's like, I mean,
It's like the name of the state.
yeah. No, this is so true.
Yeah.
But it's just like, I mean, like,
People-
I just feel like Bruce Lee, but then again, I'm from Seattle. So.
Yeah.
Bruce Lee. It's where he's buried. He's oh, okay, it's specifically a single famous Asian American.
I don't know famous. I also don't know famous.
Oh my goodness, what's their name? I mean, it's 52% of people. 9% named Jackie Chan, who's not American. That's awful. Yeah, Bruce Lee was the most common named video, which I told you. I immediately thought. So. That's so funny. It's so funny.
All right, so, whoops. I already said these things, but I don't think you all saw this slide. Okay, so anyway, these are just the few things I'm gonna go through in the last few minutes. Some examples from the literature. So, people can combine, these different approaches. So you can have an optrode, where you've got both recording and you've got laser delivery. And so whenever you have some sort of behavior, you can do that in the absence of stimulation. You can record what's supposed to be happening with your neurons of interest during the behavior. Then you can go back later and stimulate. And this can be used for two purposes. One is it serves as sort of the internal validation, like, okay, when I, stimulated, I got all these time-locked events. So I got these huge spikes whenever I stimulate it, but it can also serve, I don't particularly use optrodes, but what I understand is that, from these extracellular recordings, you can isolate single cells, and this can serve to either tell you that your laser is doing what you think it should be doing, or if it's activating some cells and inhibiting some cells, you'll have a readout of that. And it also can serve to optotag units. So you can say, okay, I put channelrhodopsin only in, inhibitory interneurons. So the neurons that were of this subtype that had time-locked activation with the laser stimulation, they behaved this way. Then cells that were negative for that responded, this way. So it kind of serves a bunch of different purposes to have that all together. And again, you can also combine with imaging, assuming that your laser and your recording wavelengths are sufficiently separated. So again, this is a case where you would definitely want to go with stimulation from crimson because that shouldn't contaminate the green recording signal. So you have to kind of think about that, which I think you covered in previous classes when you talked about wavelengths and imaging. And you can also kind of, in this case, you can appreciate that they've done sort of MRI type recordings. So they've been able to see how far the laser goes. So if you have this kind of combined recording plus opto, you can kind of see like how far is your stimulation going, what kind of activity in, you know, this big region of the striatum are you really hitting? There's a nice review if people are kind of interested in human research. You know, there's a nice review that sort of compares what's been happening in rodent literature to what's been happening with, you know, ways to manipulate human brain activity, one of which is transcranial magnetic stimulation. So, you know, the circuits generally are kind of the same. What happens if you do optogenetic manipulations in animals versus what happens with humans? So that's a really nice review if you're interested in that kind of information. So next I'm going to show you an example from a system that I think a lot of people are pretty familiar with, the ventrotegmental area dopamine neurons. We all feel good about what those neurons kind of do, kind of where they project. So I'm going to show you, I'm going to go a little bit more into depth on how this study was designed and what made it really nice and a good sort of blueprint to follow when you're thinking about designing experiments that are meant to test a behavioral readout. So in this case, they started off by doing fiber photometry, which is where you're able to look at sort of changes in genetically encoded calcium sensor activity in specific cell types. So here they were using a TH-CRE animal, tyrosine hydroxylase. So you should be having GCAMP fluctuations in dopaminergic neurons. They put the recording fiber into the ventrotegmental area. And in this case, they were looking at social behavior. So this was freely moving animals. They were not head fixed, and they were doing social interactions with another adult conspecific. And here what they've shown you is sort of the raw trace. They've represented as DF over F. So you can see sort of what's happening across a really long trial, and they've coded here in red when the animal is interacting, and then they've taken, they've magnified this so you can see sort of a specific region of the trial where they're doing a lot of social interaction, and they very nicely show that when there's these social interactions, you get sort of higher activity compared to areas when of the behavior where they're not socially interacting. So they have a recording. So now they say, okay, these neurons might be naturally involved in social behavior. We recorded, we saw that. Again, they can't tell you what the pattern of stimulation is, right? Because this is GCAMP, the kinetics are totally different. These are not electrophysiological recordings, but at least they know there's sort of this change in activity. So next they go through and they both activate and inhibit. So they're giving a pattern of light. Here they're doing 30 hertz for several seconds. Then they have a timeout period of 5 seconds. And they do this throughout like a 5-10 minute social interaction trial. And they go in and they confirm with channelrhodopsin that whenever they give a pulse, they're getting this huge response in the cells. So they've done slice confirmation that this pattern of stimulation is getting a response. And in fact, with this pattern of having it sort of on, then a timeout period, they're not getting a ton of They're not getting depolarization block for sure, so they still get a big response. There's maybe a slightly smaller response, so maybe there's a little bit of attenuation of the firing, but at least over this course, they're still getting reliable responses with these light pulses. So they're time locked, they're not like delayed. So they show that, and then they show that compared to EYFP controls, they see this big increase in social interaction when they excite the cells, which agrees with what they see here in the natural behavior. They see increased activity. When they increase the activity, they get more social interaction, and they get exactly the opposite effect when they inhibit. So they reduce social interaction. And here again, they aren't just inhibiting during like social contact. They're inhibiting during the entire trial. And I'll talk about exactly why that is the case. And then what's really nice, I think, in this experiment is that they show, every time you have something like, there's some sort of motivational circuit here, right? So are they just responding to salience or novelty, or is it really the fact that it's a social object? And so they show between halorhodopsin that there's no difference in interaction with an object. So this appears to be specific interaction with a social target as opposed to just an inanimate object. So putting activating these VTA neurons doesn't just make it more interesting to explore something near you. It was specific to a social target. So I thought that was nice. They also did some sort of circuit mapping. So here their same VTA stimulation. Then they looked at a couple of projections. So we know where these neurons should be projecting to. What happens in the downstream areas when we give this VTA stimulation that now has been correlated with increased social interaction. So in this case, they did the same kind of stimulation for a couple of minutes. Then they waited 90 minutes, and that was enough time for CFOS protein. So we talked a little bit about that during the active cell labeling. So then they looked to see, okay, which downstream regions is this kind of stimulation leading to more activity in? They saw increase in CFOS positive cells in the nucleus accumbens. They saw an increase in CFOS after stimulation in the prefrontal cortex, and there was no significant difference in CFOS in the basolateral amygdala. So then, now they have these two different projections. So they go and they look and see what is the effect of these different projections on this social interaction. So they show that now if they move the laser to the nucleus accumbens, they're getting this increase in social interaction. But if they put the stimulating fiber in the prefrontal cortex, there is no increase in social interaction. They go on further so there are some caveats with just moving the laser. So if you stimulate one projection of a cell and it projects to all these different places, there could be back propagation of action potentials. Did you all cover that sort of in like part one maybe? So this can happen. So essentially you could get exactly the same effect of stimulating in the PFC as you do in nucleus accumbens, because you in effect may be stimulating everything when you stimulate one thing. And of course, axons have like really specific sort of projections. They have really unique physical properties. So some projections may do the same thing, some may not do the same. You may get back propagation to some of them, but not all of, depending on how it gets to the soma, whether there's inhibition in the downstream, there's all kinds of circuit effects that could contribute to back propagation or not. But it already looks good, right, that they have a different effect. And so they also go in They have now done VTA in, canardoxin in the VTA. This is stimulating everything. So we already saw this data. There's an increase. Now they've moved the fiber to the nucleus accumbens. Now they've used a method where they've put a virus in the nucleus accumbens and they're stimulating back at the VTA. So this was only infected through the terminals, and then they inhibit, ensure that they get an opposite effect. So they kind of independently confirmed that this projection could be having an effect on social behavior. So that was a nice control, doing a different type of projection stimulation. The other thing that they showed is that they have this change in social interaction with the nucleus accumbens. Oh, great. So there's no effect with PFC, but like surely stimulating the PFC does something, right? So what they found was that there was a big change when they stimulated all of the VTA neurons in this open arm time in an elevated plus maze. And what they found was that that effect seems to be mediated by the prefrontal cortex. So this phenomenon of saying like this projection does one thing and this other projection does something different. They don't do the same things. It's not redundant. They're actually specialized. This is known as double dissociation. So this is also a nice thing to be able to include if you are comparing and contrasting the function of multiple projections. So that all comes from, again, this Cell paper from Lisa Ganadin in 2014. And then the last thing they showed is that, okay, these changes in EPM, these changes in social behavior are not because they just move more, which again, you know, you have this cell population that is involved in movement and motivation. And it's not just some general novelty effect, and it doesn't appear to be mediated by locomotion. So they really nicely go through and dissect out what is this one pathway involved in, what is the other one, and what behaviors are they not involved in, to show that it's not some sort of global phenomenon, that all these different things are changing. It's really specific to social behavior for nucleus accumbens projections and anxiety-related behavior with the prefrontal cortex projections. And this all tied back into the original reporting.
How is doing all VTA and then just VTA to MPFC? How does that show that only the projection to the MPFC does the elevated plus VTA?
I don't remember off the top of my head if they also show that nucleus accumbens doesn't, but I think that probably is in there. But I don't recall.
Oh,
but
so
I wanted to demonstrate this double dissociation phenomenon.
right, so you'd have to do like all and then the different projections.
Yes,
right?
yes. To have kind of like a matrix of
Right.
like check, X, X,
Yeah.
X, check, you know, that kind of thing. So I think that's a really nice kind of roadmap to follow and just kind of give you some real life examples of some of these concepts. What is it that I'm talking about when I say depolarization block? It basically means, again, I showed you an example already of where the cells couldn't follow certain trains. They had a huge effect with the first one and then never responded again. This can happen. So here they show that with certain pulse widths, you start to get depolarization block. So here, every single time you have the stimulation, they can respond. But if there's not enough recovery time between, you can go really quickly into depolarization block. I believe these are different projections. So in one case, it's the medial habenula is these two with the different pulse widths. In this case, it's these projections to the striatum. And I don't remember the exact cell type. We're not talking about VTA neurons anymore. This is some chat neurons. I forget where the cell bodies are exactly. But basically, these show a depolarization block after a certain amount of time. So they can follow for a little bit and then shut off. And so you don't know if that's happening unless you've done some recordings. And in the same paper, here again, they're comparing a bunch of different, so these are a bunch of different Cree lines where they've expressed channel rhodopsin virally with CRH in the olfactory bulb. They've done different light pulse durations, and you can see the black line they can follow for a while, but eventually they'll go into depolarization block. And the curve always they always go into block after some amount of time, but the actual behavior of these different cell types, where they're stimulating, whether it's cortex striatum or medial venula, there are differences. So these are really specific, again, to the reagents that you're using. So are they chat cells? Are they VGAT? Are they VGLUT? Where do they project, et cetera?
Is that also something that you would find like in literature is like you need to use this for stimulation to avoid depolarization?
You might, or you could at least find, you may not find, a nice curve where it shows like, oh, this is where it goes into block, but you may be able to at least say, I'm going to use this because it's been previously shown that this does not cause depolarization block, right? So you might not have this nice like curve for everything. So another caveat is disruption of GABAergic signaling. So I think you probably talked a little bit about inhibitory opsins. So there are two big classes, although now there are more, but as of a few years ago, these are the ones that were predominating. So different ones may work through slightly different mechanisms or with slightly different kinetics. But halorodopsin and variants are chloride channels. So they pump chloride in, and that hyperpolarizes the membrane. But in the case of archaerodopsin, it actually pumps protons out, and that's what causes inhibition. That's what causes the hyperpolarization. And so, not to get into too many details, but essentially what you can see is that, okay, you get this hyperpolarization here, and as soon as the laser goes off, there's this giant spike, okay? So that happens immediately after the laser turns off, and that's why, and to some extent, it happens a little bit with halorodopsin. You can also get like some kind of weird stuff, like right at the beginning of the laser. You don't get as much, at least what these people showed, comparing, halorodopsin activity to archaerodopsin activity here. But you can see here in this example, depending exactly how much power they were giving, they could also see it with halorodopsin. In this case, they saw a big downward deflection with halorodopsin with some of the different powers. Whereas when they do archaerodopsin, they actually get a deflection in different directions. So when the laser comes off, all this weird stuff starts happening. So that's why it's really difficult to do a time stimulate, like a pattern of inhibition. So with excitation, you want it to be firing. But in the case of inhibition, you're going to get this weird rebound firing potentially. And the different variants have different kinds of rebound after inhibition. On the other hand, this could be seen as a feature instead of a bug. So in this case, people were actually taking advantage of the fact that halorodopsin showed this huge rebound firing. So it's actually, depending which opsin you use and what kind of readout you're using, essentially they're all going to do this. And it has to do with the fact that you've hyperpolarized, and as soon as you stop pumping either chloride or protons, whatever direction it is, there's going to be this huge shift back to equilibrium, right? So in this case, they actually took advantage of that to cause seizures. They were doing inhibition in this pattern specifically to cause seizures, to get network synchrony. And again, we've had some nice whips recently that show, these different power bands can show synchrony between regions, and they harness this weird thing to actually induce seizures in this neuron paper from 2017. So that's sort of the biophysical reason why inhibition really has to be throughout the whole trial. But you could keep recording after and see if something weird happens as soon as you take the laser off. I guess that could be a nice internal control. So another caveat is that when you put the laser on, you're kind of taking advantage of the fact that it stimulates at a certain wavelength, right? We think of it as putting in 488, 532, depending on what the option is, right, to trigger it. But it also changes the temperature because that is light, and light creates heat. So in these two different papers, they show you what happens to the change in firing simply due to changes in temperature due to laser introduction that has nothing to do with chanorodopsin or halorodopsin or archaerodopsin. So there's not even an opsin here. It's just showing you that the firing rate will change based on the power of the laser. So this is why I mentioned the thing about really titrating down that laser power as much as possible. So that's going to lead to minimal heat artifacts. So you might be exciting cells in your YFP animals, even though there's no chanorodopsin there. So that's why that's included as a control. And so, yeah, so why you want laser off and laser on, as well as this external YFP group. And again, we've already kind of touched on this with the, rebound firing that happens, within the region you're looking at, regions outside. So this is a nice example. I'm not sure why the bottom is cut off. I'm sorry. Oh, okay, good. At least the citation is here. So we'll have to check that slide. But here, they're actually stimulating VIP neurons. So these are inhibitory interneurons in the cortex. And so when they do that, they get this really nice time-locked activation of VIP cells. Some cells, they're just recording all kinds of activity in the cortex. So when they do that stimulation with the VIP cells, where they get nice light entrainment, so to speak, some proportion of those cells have, like, within 200 milliseconds show this inhibition, whereas some of them show this delayed activation. And so those could be projection neurons going out of the cortex that stimulate other things downstream. So even though you think you're isolating some behavioral effect of VIP neurons, you have to consider that that now leads to maybe synchronous firing of excitatory cells that are involved in something totally different. So, and these delayed activations could look different depending on the downstream region. So here they looked at effects on firing rates in the auditory cortex, as well as in the medial prefrontal cortex. And you can see here, there's kind of this long increase in delayed activity, whereas in the medial prefrontal cortex, there's more of a narrow effect. So you have sort of these weird circuit effects when you do these kinds of manipulations. And this is the kind of thing that you might want to address in the pitfalls and alternatives. Okay, I did this really specific manipulation, and it may have had effects on other behaviors because of the projection patterns and effects on other brain areas. In addition to the heating effect, you can also have sort of noise if you are recording. So the heating can change the firing of the neurons, but even in this dish where there's no brain, there's not any tissue here, there's no cells to fire, the recording electrode is picking up activity just because you shine the light. So this is known as the photoelectric effect. And so this has to be taken into account. And then this speaks to the question about what kind of pulse trains you might want to give. So again, these are comparisons between a virus where AAV1 was expressing chanorodopsin non-specifically, and then Thy1 mediated channelrhodopsin expression. And so you can see when there's sort of like widespread expression of channelrhodopsin, too many cells now seem to be sort of expressing this channelrhodopsin. And even though the cells themselves should be able to follow this, something about this viral injection, there's too dense of an expression of channelrhodopsin. Maybe there's too many inhibitory and excitatory neurons that seem to be sort of interfering with each other. You get this sort of loss of being able to entrain the stimulus. Whereas if you have a lower density expression of channelrhodopsin, because now you've selected specific cell types, the Thy1 channelrhodopsin seems to be more like doing an electrical stimulation, is able to follow that train. And so these types of behaviors can be different depending, again, if you have different frequencies. So there's a slightly different effect of 10 hertz stimulation compared to 50 hertz stimulation. And then in the example, I don't know if this is the right or left for you, but anyhow, over here. These are the newer different types of channelrhodopsin. And again, there are different ones for inhibition now as well. So these all have different kinetics, different wavelength sensitivities, etc. So the behavior of them across different stimulation patterns is a little bit different. So at 5 hertz, if that's your stimulation pattern, you don't need to worry too much about choosing a specific opsin. They all follow it. If 10 hertz is your choice, you probably don't want catch. That's going to go into depolarization block. This one will as well at some point. Maybe chief would be better. For 20 hertz stimulation, chief may be a little better than some of the other ones. So you just want to really match the duration of the stimulation, the frequency, and then the timeout period to see if they can recover. So obviously, none of them really seem to be able to follow 60 hertz for very long. Probably that's way too fast. I've rarely seen something over 20. I typically see 10 to 20 in papers. But there are some of these good biophysical property methods papers where if you're trying to choose one for a certain application, certain types of behaviors, you can match the properties of the actual opsin. You also need to know what the neuron can do, because the neuron itself might not be able to follow certain stimulations. So matching sort of what your stimulation pattern is to the opsin, the neuron, the behavior, all of those are important to kind of keep in mind. And again, it's the kind of thing you only have to explain once in a proposal. Why are you choosing this one? For these reasons. And then here, this is related to laser power. I can't think off the top of my head if I've seen this. There might be something nice. This paper is a nice comprehensive technical paper, Jackman et al. 2014. So I don't recall if they did different frequency patterns, but at least as far as laser power, they showed that they really needed a lot more power at the axons compared to the soma, and that's probably due to the copy number. Because again, axons are pretty far from the nucleus. If you're using a Cree-dependent recombined expression system, that Cree is coming from the nucleus, so anything that's coming from that has to be translocated. And just to that point too, if anybody's really, really interested in projections, either recording using some viral, GCamp, or using channelrhodopsin or halorhodopsin, you need more time for the virus to get down there. So if you say, oh, I'm going to wait 2 weeks because that's how long it takes AAV to express, you might not see anything. And this goes to, even if you're using something for anatomical tracing, or if you're using retrograde AAVs, you need more time for something that's axonally taken up. Or if you want something expressed in an axon, it's going to need more time. So usually on the order of 4 to 5, 6 weeks for expression. So you may start stimulating and you get nothing that first week. And then suddenly 2 weeks later, you do it again. And now there's this huge effect. And it's probably because the virus construct simply wasn't at the terminal at that time. And then here are two papers that kind of have sort of conflicting results, but just something that you kind of have to keep in mind. So in one paper, they compared and contrasted. So before I was just showing you about this rebound activity with archaeorhodopsin and halorhodopsin. That was really from the soma. Weird stuff might be happening at projections because of the nature of these different channels. So in this case, they were using a GCamp to record activity at projections. And they found that when they stimulated tried to inhibit using archaeorhodopsin, they actually got increased calcium at the synapse. And so that could lead to neurotransmitter release and effects, which is the opposite of what you're trying to achieve if you're using archaeorhodopsin. And what you can kind of appreciate is that it's not as if halorhodopsin does not have this effect. It's just less compared to archaeorhodopsin. So again, in the summary traces, you can see the red is archaeorhodopsin, blue is halorhodopsin, on, off, on, and you get this huge increase in terminal calcium influx when you use the proton pump archaeorhodopsin. But then when they looked in slice using archaeorhodopsin, they said, oh, it's just fine. It's all dependent on the laser power. So you might actually get outward activity if the laser power is too low, but as you increase it, you get better inhibition. And so, there may be some technical ways to overcome this weird stuff, or you can just use a different opsin. I think there are newer ones that are probably better. One, if this is something that you are going to encounter, I will say that people really love dreads at terminals because it's a G protein coupled mechanism. It keeps activating because it's an intracellular signaling cascade, and it stays really local. If you're locally delivering CNO, it stays at the terminals. So, but you do have to do some sort of cannulation to be able to locally deliver that dread agonist. But there are some cool little tricks that you can do. So there's, I've been talking a lot about some caveats. There's some cool things you can do too. And so in this paper from 2014, they showed that if you have behavior that is significantly changed by cueing a shock, you get this change in lever pressing. And you can actually mimic that by simply stimulating the laser in this, I forget what brain region we're in. I think this is somewhere in the hippocampus. That you can just stimulate some neurons and, oh, it's lateral, it's right there, lateral amygdala. So you can pair laser stimulation of lateral amygdala neurons with this shock, and you can essentially get the same behavioral output. So in this case, there was no cue, you just mimicked some sort of pairing with this shock, and you could get the same behavior output using optogenetic stimulation. So that's sort of like a LTP, supposedly, you have these coincident signals, and it can either be behavioral or simulated neural activity. And in this case, they did sort of the same type of thing where they had an optical fiber in this circuit that mediates ethanol intake. So after LTP, they could get this enhancement of ethanol intake, and that did not have an effect on sucrose, so it was really specific to alcohol. And then if they delivered a pairing that was long-term depression, they could change behavior that way too. So if you're interested in sort of doing these sort of optogenetic manipulations that sort of pair with some kind of other behavioral parameter. These are two good recent references that have done that. And this can be, you can potentially think about this in terms of state-dependent plasticity. That's kind of what we think about when we think about alcohol behaviors, sort of a state. Disease-related plasticity. So we did show you can get these seizure-like states using this entrained inhibition. And then you can also think about, maybe I can induce homeostatic plasticity using a certain type of stimulation, see what happens. And then there are some brain regions that laterality really comes into play. Cortex is one of those areas. So this is really something that's very, very important in human studies. Of course, we know that there's a lot of lateralization in terms of language and other types of behaviors, particularly that are related to the cortex. But a lot of times we don't really think about that in animals, even though it might still be important in certain behaviors or certain brain regions. And so in this paper from 2016, I don't fully understand all the details because I'm not a delay match to sample type of person. I'm not a cortex person. But essentially what they showed was when they did a unilateral stimulation with chenoredoxin, they got this particular effect on behavior. And then if they did unilateral versus bilateral, they got very different effects on the behavior. So you can see these results look a lot different from the unilateral. And then they were doing recordings and showing what the behavior response is. So these are behavior raster plots. These are spikes in the regions. And what they found was that ipsilateral, contralateral, or bilateral stimulation patterns resulted in really different firing rates. So this is something to keep in mind if you're doing. Typically, we do bilateral stimulation if we're doing something because we don't want to have some weird biased behavior result. But there could be something interesting if you do one or the other. And then the last slide, sorry, I feel like I'm going a little over here, but the last one is just to speak to the neurochemical complexity. And again, going back to sort of dopamine, striatum, we know that there's all these different neurotransmitters that may be co-released, other peptides. So here and again, part of the reason that I'm showing you real examples is like, this is now it'll be on Canvas. If you're writing a research proposal, just go back and cite these. Again, like I said, it's all about making a really clear, concise statement and then putting like 10 citations. Okay, so you go, oh, these neurons could be complex neurochemically. These 2 citations, I showed it to you in class. So we might want to go and do an ePhys experiment and show. So what they did here was they had VGLUT2 expressing cells and VGLUT2 not expressing cells. And they see that if they stimulate different cell types, they get a really different effect based on the projection that they're looking at. So if they stimulate the VGLUT positive cells, they got this EPSC in the nucleus accumbens shell, but the same type of stimulation at dorsal striatum did nothing because those projections maybe weren't going there or there was a competition or something like that. So you get really different effects, the same stimulation, same cell types, just different projection target areas. So you can't necessarily generalize like, oh, I got excitation here, I'm definitely going to see it there. They may not go there. And then in this case, they showed that there was a mixed effect using slice physiology. They had these recordings in, I think it was striatum, I forget the details, but they can show that this optogenetically stimulated activity was sensitive both to gabazine, which is a GABA antagonist, GABA receptor antagonist, or to MBQX and CPP, which are glutamate receptor antagonists. So you had a mixed effect here, and they went through and did really nice dissection that the EPSCs were sensitive to like calcium mimickers or The glutamate receptor antagonism and the iPSCs were really sensitive to GABA antagonism. So just showing that, you know, these different things were being released when they stimulated these dopamine cells. So just some citation. Feel free to use this, like I'm telling you this because you should use it. It's not like my intellectual property. Please get F31s and pass your qual. Okay? Sorry, I know we went really fast at the end. Questions, comments? All right, so that's our last class. The next time we meet will be for your final on May 13th. And we are meeting in this same room, 1:30 to 3:30.
But I wasn't, but yeah, I did the like, oriental awards. Honestly, the stuff about something so cute. I saw it on my proteins. Somebody had it, like, candy.
I don't know why I thought she would be back for the spring, like, I totally forgot about that. I mean, Neil forgot, or he didn't realize there was nuts. I felt so bad. Oh, no. Poor Neil. So, did you get my email?
I received it. I did not read the whole thing. I'm still preparing
Totally
for
fine.
class.
That's so totally fine. I can just like, I guess, talk about the like changes.
Yeah.
So we had meant instead of, we made a silly little mistake. And instead of putting.
I don't know the acronym, so that was probably not.
Okay, well, we meant caudate instead of putamen, but
Okay.
dorsolateral striatum, putamen. That's why. Miscommunication, but,
Yeah, I think the main thing is like, is it projections or
Yeah,
cell bodies or whatever?
yeah. So. So I think we have like too many questions. And I think we